Hello everyone, thanks for coming. Excited to introduce today's speaker, Wallace White. Wallace has over 10 years of really great product development experience, ranging from areas such as medical devices to consumer electronics. Currently, he is the director of point-of-care diagnostics at Stratos, where among other things, he works on the Bill and Melinda Gates Foundation's project, the PanDx, and he'll be talking a bit about that today. So please join me in welcoming.
Thanks, Tim. Well, thank you very much for having me. Very glad to be here. Nice to be back in the Bay Area where I used to live for a while in college. And being here in March is like being in Seattle in July. So it's a nice taste of summer almost. This is my favorite time of year here too. Everything's still so green. I know we're supposed to like the Golden State, but it always looks kind of brown and by the time summer will go around. This is just paradise. So I'd like to talk tonight about the development we've been doing for this project for a few years now for the Gates Foundation, this PanDx project, as we call it, to make an integrated diagnostics platform. The main content of the talk tonight is actually going to be about the experiences we've had and things we've learned from traveling around the world and going and touring health clinics, meeting with ministries of health, and keeping in with peers, professors, and attending conferences, those sorts of things. At the end, I'll give a bit about the technology of PanDx itself, about the main project, where we're in some sensitive talks right now with potential commercialization companies, diagnostic vendors for that, so we can't go into too much depth about that. So please forgive me for that, but I hope that the content, talking more about the stories and the places and the experiences that we've seen will be very interesting to you. I hope so. So I'm glad to have questions at any point during the talk. If I don't see you, just give a little, either raise your hand, say something briefly. I don't mind interruptions. I'm known for having kind of narrow vision, so especially if I don't see your hand, don't think that I'm ignoring you. So first, a little bit about Stratos. Stratos is up in Seattle, Washington. We're a firm of about 90 people. Today, it was started in 1987 by a mechanical engineer, an industrial designer, and eventually a WE electrical engineer as well from the Food Corporation. So all guys that worked in test and measurement, guys that knew about designing rugged products and worked in a very mature product development cycle there, decided they wanted to make their own thing and start focusing on that for lots of other companies. So over time, it's built up into the size that it is now, where we are a group that can take on engineering and product development challenges for anybody around the world. A great number of our clients are on the West Coast, but I'd say the US represents a great majority of our clients, but we do have an occasional client out of Europe and other parts of the world as well. We're all in one single office. We have a nice integrated environment where we're mixed up between all the different disciplines that we have. We don't have a mechanical engineering team or something like that. We intentionally promote mixing and interaction between all the different groups there, because the next great idea might come just as soon from talking to somebody who has no expertise in your field, but has their own perspective about it and can inform you, help you think of something new. Part of why we actually really like diagnostics and the life sciences projects is it's fun to have people from so many different disciplines who you have to bring together, because very few people know all the things they need to know to be able to make these kind of products. It's exciting to see that intersection where you get those different expertises together. So there are the different disciplines within our group. I won't go on too long about this. I'm going to get on to the good part, talk about the project. We work with a lot of life sciences companies. That represents about 3/4 of our business in total. So Vitro Diagnostics is probably 25% of our total business flow and has been for close to 15 years now. Working with microfluidics companies, glucose meter companies, a range of things, and people who are bringing diagnostics closer to the patient. We do stay involved with consumer electronics, and as a firm, it's a good thing for us because it keeps us aware of new technologies. So things we learned about the consumer electronics project a few years later are ready to go on to a medical project often. And then a lot of clients, of course, also are very aware of consumer electronics. And if we had a penny for every, a nickel for every client who came in and said, I want this medical device, I want it to look like an iPad, or like an iWatch or something else, we'd be very rich. That is where people are today. You work with these great consumer electronics, you want to see your medical devices be just as great. Before getting into the PanDx story, one other global health project. We've worked with Global Good, a group out of Redmond as part of the Intellectual Ventures Laboratory, on developing a device to be able to meet the cold chain need. So we're getting vaccines into places where you need to keep the vaccine cold for it to be stable and still have efficacy. But there's no electricity available. So think of remote areas of Africa, places that might be a couple of days' journey away from a major city. There are products people are working on to make solar refrigerators. At least some of those actually are on the market, I should say. But the folks at Global Good started thinking that a different way with encouragement from Bill Gates to look at this problem. They said, how else could you do this? They came up with the approach of basically a super thermos. Take a dewar like you might use for liquid nitrogen, reconfigure it to be able to do something that's just a few degrees above freezing, and make a product out of that. So we worked with them after Global Good had spent years working on this and done many iterations of it, done a lot of the basic science of it. They brought us in to help turn that into a product so it could actually go into a place like this. So it's the barrel-looking thing you see at the top left there in a site that's being used for vaccinations. So this product overall really is a super thermos. It's a dewar, you might expect, with different installation techniques and a lot more ruggedization so that it can actually go into the field and be used in these places. We also designed for them an electronic system so that it monitors the temperature of the device. It reports back to a central site where it actually is with its own GPS device and with a GPRS radio so that it can be tracked so it can be understood during the development. But the whole thing runs off ice. We call them ice batteries sometimes, ice power. The whole idea is that these blue slivers that you see here, fill them up with water. It's a plastic jug, just fill it up with water, throw it in the freezer at the base station where you do have electricity in a larger city. And then take those out, drop them into our thermos, and you're good for a full month. So you keep drawing vaccines out each day, but still keep it below 8 degrees C for a full month. And it's done quite well in trials that way. So we worked with Global Good and built, I think, about 15 prototypes that they used for field trials a couple of years ago. And they've since gone on to work with Aqua, a Chinese manufacturer, to build this into a production device that's ongoing. Some of the things that we found that were interesting from doing this, we did have one of our guys went along to Senegal on one of the trips that Global Good did to see firsthand how the rollout would go. And we spent a lot of time worrying about how rugged this device would be. We knew that it would be transported long distances over very rough surfaces and in lots of different ways. And in particular, we were prepared for being strapped to the back of trucks and sporting utility vehicles for that. So a lot of shock and vibration work. A lot of consideration of what if this thing rolls off the back of the truck to make it strong enough to withstand that. And we did very well in all those tests, all those preparations. And then as things went along, then we heard later from Global Good that they'd done a study in far Ethiopia where it was a couple of days journey by camel to get there to this community to do vaccinations. And they got it there, and then they showed us how they got it there. And they strapped it to a camel. It's a natural thing. You shouldn't have been so surprised, but our guys saw that and they went, oh my gosh, it's not standing upright. They'd always designed the device to be standing upright on things and realized we really had overlooked that. We thought about hand carrying, we thought about putting on trucks, different vehicles, but not about being strapped on a camel. So if we had anticipated that, if we'd been out there earlier and discovered that, I think we would have designed either a special harness or maybe some different handles on it to be able to keep the orientation the right way. Because the thermals actually work better if it's upright. The vaccine bottles and the containers don't rub as much. Those kinds of things. But it was a great experience for our team to get to go and see that. Another thing that they brought up was that with pulling all these ice blocks and needing to load them into the device, we've done all our work in Seattle within our offices and places like that. And then they got there and did the work there and realized a lot of times you've put this device on something that's not quite level, and you've got these ice blocks that are very slippery. So trying to stack up ice blocks inside the device, or just setting them on a table somewhere to work, and there they go sliding around the table, that sort of thing. So it was early enough that the team was able to build in more interlocking features between these blue plastic jugs to help them to lock together better. It's one of those things that's kind of hard to anticipate. Even as hard as you might try, and talking to people who go to the field, there's nothing quite like actually being there and seeing for yourself what your concept does with people that are really going to use it and what they think of it. At the Grand Challenges meetings, the Gates Foundation and others host each year. This last year, Bill Gates spoke about that, about the importance of, more than anything else, actually going there to talk to the people, get to know them, see what their lives are like, see what they do from day to day, see how they're doing the thing now that you're trying to help them do. You just can't do it any other way to really do it well. Another great thing was this quote that our guys heard from them. Please thank your teachers for educating you to be able to do this work. I think sometimes it's easy to think of yourself as the hero here, that you're doing this great thing for people who save the day, save the world. This was a good, humbling thing to be reminded of, that not only should we be thankful for all the people who helped us to get to where we are through education at places like this great institution here at Berkeley, but all the people who have contributed funding, contributed support, all the many advantages that so many of us have in this life that have enabled us to live the lives we do today. To be thankful for that, and to be thankful for the chance that we get to hopefully contribute to improved standards of living, improved welfare around the world through this work. PanDx. So Stratos, for many years, have been developing diagnostics for commercial clients, people who are concerned about delivering better tests in the US and Europe, coming up with something that was faster, a little cheaper, a little more sensitive, a little more specific, something that was doing better things that we were already doing. Not very often, frankly, to get to someone who's being untreated today. There are cases where point of care in the US and Europe are good for that, where actually you can say, we're going to catch a patient who otherwise would not be into a health center often enough. We can catch them, we can then provide a treatment on the spot and prevent transmission further. It doesn't happen that often. When you go to areas around the world, there's really a different mindset there. There's a greater opportunity there for how much diagnostics can do. So we've taken the experience that we had through years of ongoing consulting with these clients and then taken that towards the developing problems thanks to the development of the Gates Foundation, bring us into this. Bill Gates summarized on his blog recently the basic idea of our little project, which is, can you create a new device that quickly diagnoses HIV, TB, malaria, and other diseases, accepts different samples, like blood, saliva, and sputum, is affordable, reliable, and will work in a small clinic that only has a few hours of electricity a day. We're not trying to solve every single kind of diagnostic situation there might be in the world. We're particularly not on projects trying to go down to what we call a level 0 health center, to the individual with a backpack, or to the roving health clinic worker, or even the small health post that's a single room. We're actually not trying to address that need in our project. The Gates Foundation pointed us towards the health clinic, a place where you already do have clinicians, you do have staff, you have patients who would come and seek care, but diagnostics are scarce. They're very limited, or they're either can't provide a very high functionality test to really help people, or it takes too long, either because it's a slow test there, or that it has to be sent out to a centralized facility somewhere else. So we were excited to see that Bill Gates went on and mentioned the effort that we're part of and showed the project that we're developing for their company at this. We actually hadn't been authorized to make any kind of public statement about what we were doing until this. And, hey, that's great, Bill Gates said it, so I guess that's okay for us to talk about. But since 2011, our company's been involved with the foundation, traveling around the world to better understand the needs for diagnostics in global health situations and coming up with concepts. It became a bigger project about a year and a half ago. We'll talk about it better. But first, let's talk about those roots, things that we learned through that. So we traveled to 6 countries that is what Stratos staff have, including Brazil, Peru, Kenya, Ethiopia, South Africa, and India, all for visits to health clinics. Some for a couple weeks at a time, some for India for a little bit less while we were there for grandchild meeting. And we've had meetings from directly with laboratorians, as you see in the lower left there in Brazil, to meeting with the head of the National Laboratory Service in South Africa, you see in the middle picture, and meeting with clinicians, doctors, nurses, everybody along the way, and health experts at universities in these places too. And it's been a hugely informative thing, a great experience. So I'd like to go through a number of things that we have learned. I wouldn't call these lessons learned exactly, but at least topics and observations that I think are worth sharing. So the first one is the importance of complete solutions. It's pretty obvious in a sense, but I'll go through an example. How important it is to have not just a technology, not just one thing, but to actually be able to deliver a complete solution that results in better patient outcomes. When our team went to Kenya and Ethiopia, they came to one of the health clinics they were visiting. Going to health clinics day after day to understand what kind of tests they perform today, what their available infrastructure is in terms of power and water and what temperatures they experience, things like that. And when they show up at one of these, they see this crate sitting on the front porch of this health clinic and start asking them, so what is this crate? It's an Abbott Celodyne. It's a really nice hematology machine that was donated and sit here. So, what's it doing out here? Well, we don't have the right electrical hookups to be able to use it, so it's stayed here. It had been there for more than 6 months on their front porch, hadn't even been opened. And unfortunately, this is a fairly common story. If not exactly this happens, it's the case that donated equipment is sent and is used for a very short time, but then the first time that it needs to repair, there's no one available to repair it. There's no one who knows what to do with it. Sometimes the service manuals aren't available in local language. So often there is an equipment maintenance room or building at one of these health clinics around it, which could more or less be called a graveyard of equipment. So a lot of things never come back out of it. So see, it's really important that you not just have technology, but that you have a complete system that can enable its good delivery. You need a service and support infrastructure. You need people who understand the local situation to be able to assign the right equipment to the right places. And a team that can help ensure that it's used well. And my company can't do all those things. We're depending on other groups that do that, working with other groups and with the Gates Foundation and other funders who are working on completing that whole ecosystem of good testing. Second topic, flow. So just north of Lima, Peru, in the city of Callao, is where you see a lot of peri-urban areas that really represent where a lot of people, a lot of the impoverished people today live in the developing world. These are places we've been seeing. We tend to think of communities like I showed in far Ethiopia, where people living truly in independence, a very small community or tribe somewhere. But there's a lot of population now that lives close to a big city, but is out on the fringes of it, lives in informal housing somewhere. Shanty towns that have been built up without proper ownership and built up very quickly in the last few years as people have come out of the more rural areas and come into town seeking work. So this is one of those places that didn't exist 10 years ago. We went and visited their health clinic there. It was actually this was one of their larger clinics in the area. And we talked to them, said, explain to us what the patient flow is through this laboratory. How does it work when someone comes in? What do they do to get a diagnosis and treat this person? Well, on day one, you come in and you wait in line to get a ticket to be able to go and see a doctor, see a clinician or a nurse about it. So a lot of standing around, a lot of waiting rooms for this. And you go through and get your slot sometime later that day. The doctor looks and says, okay, yeah, it seems like you might have tuberculosis. Let's go and get a sputum sample from you. Come back tomorrow. We do sputum sampling and we do blood sampling in the mornings. So do that the next day. Day 2, patient comes back, and this is another day of work, spends a significant portion of income perhaps to be able to get there, and provides a sample during certain hours. The laboratory may assess that sample that day. They may need to transport it to another site somewhere else around the community, or they may just have a big enough backlog they don't do them all that day. So day 3, in the best case, but often much later than that, the lab actually runs that test. So they have these big piles of samples stored up from their clinic and from other reporting clinics. And they would go through and do a big batch of those tests. So just doing TB smears for 3 hours, or run all those, and I'll do my malaria test later, another test later on. So finally on day 4, the patient comes back for a 3rd visit now for this, to hear, okay, what was the diagnosis? What are we going to do about it? So 3 days of lost wages, 3 visits, 3 flows through the waiting rooms, through the receiving, through all those people. And I went back and talked to our manufacturing guys our company and said, I think there's something to be said here for lean manufacturing. What should we be doing here? And they look at it and say, all those samples, look at that, it's just all that inventory of things that have queued up. A lot more than that in some places. Manufacturing loves to get things down where you have minimal inventories, where you're moving things through, there's minimal waste, there's minimal delay, there's minimal cost built up in those pieces that are waiting around. In healthcare, you can also look at all the samples and think, every one of those is a patient that might be transmitting the disease right now to other people. And tubes just sit there. And we haven't told the patient to do anything differently or treated them. Or if we have, we did it without really knowing whether they were sick or not. We might be promoting antibiotic resistance. So really when we talk about a point-of-care instrument, something where you could get a test quickly, we're talking about a pretty big change in how they do the workflow at these sites. They're set up almost like amusement parks in some places where there are turnstiles and there are serpentine things like, I guess like you might go through at the airport when you go through security. Area after area is set up for managing these queues of people. Again, the manufacturing people look at all that queue. This is terrible. Let's get this down to just moving people through the system. Let's be respectful of their time. Let's help them get back to work. And let's get them treated quickly. So the idea is, day one, you do it all. You get treated, you go. So it's just, sometimes it's called test and treat. That's the dream. So number 3, multiple perspectives, multiple customers. At another South American hospital that we visited, we got a good tour. It's a very large site, much larger than we're usually focused on for these kinds of products. So we're trying to really bring more advanced tests to places that have very limited testing today. This is a hospital that we were told had the best lab in the country. You should go and see this, just talk to them about your project, get their opinions about it. So we went through their labs, and their labs were a whole wing that was about the size of the building we're in right now. Nice setup, plenty of automated equipment, some manual, but fairly sophisticated equipment. And we talked to them about our little system that we were developing. And we got very little interest. No, we do all this now. We've got these machines that do these things. We run this many samples. Yeah, our problems are these other things. Bureaucratic. We've got a very long lecture about the difficulties of getting good tests through the supply chain. But they just didn't really see how our little system would fit in. Didn't seem to really care that much. But then as we toured around the hospital more, we kept encountering people who said, oh, that's what you're doing. You should go over and find this room. Because I think over in the neonatal ICU, I think they're doing something kind of like what you're talking about. So we got there, and looked around, and the doctor there, and the nurses explained what they do. And explained it for blood gas analysis and for clinical chemistry. They got their own Roche machines for it. We got a nice Roche Coloss B223, I think I've got that number right, and a Roche Ruffitron. And the nurses run it right there on site. And we're thinking, how come the guys back in the central lab didn't even tell us about this? And they got their own funding for us. They did it completely on their own because they said, what we need to take good care of our patients is we need the testing quickly. We need it right here. It's our responsibility to care for our people and do it well. This is the way it should be, they said to us. So you have to think about the perspectives. Yeah, sorry?
Do you think there was hospital politics involved?
I think that's fair to say. There are control issues a lot of times.
Oh, yeah.
About who gets money for certain things and who is the expert, whose domain it is.
Okay.
So we did hear in this situation that there were some things where the central lab actually wanted to get responsibility to be able to take over this site, and a small lab that was also next to the emergency room.
I see. Thank you.
So it's, when you talk to people who are laboratorians, oftentimes you do find they've got a lot of vested interest in the way they do testing now. And when you say, I'm going to bring this great new system to you, some people get excited about it. Some people see the patient benefits for it. Other people say, you're automating my job. You might be taking my job away or making me less important for expertise I've got.
Yeah.
Which is a difficulty. It's something that has to be considered.
Thank you.
Yeah, thank you. So, going back to multiple customers, multiple perspectives, it's good to talk to lots of people in lots of different parts of any system that you're trying to help, trying to understand and design solutions for.
Wallace? What's that picture on the left?
That was the kind of receiving area of the waiting room for the neonatal ICU. So the middle picture there is a view into the room with incubators for premature babies. There were probably 100 incubators in this hospital. It's a pretty sobering sight. For in diagnostics we trust? Question mark. So the first international trip that I had with this project was to New Delhi, India for a Grand Challenge meeting in 2011. The Gates Foundation hosted. At our hotel, every time you went in and out of the hotel, you had to go through their security standard. Any kind of backpacks you have on the metal detector, on the conveyor style thing like an airport, walk through the gate. Every time I walked through it, it went off. It buzzed. And the guy said, oh yeah, wave me on it, I just went off. Have to think about what people actually do with the information they get from diagnostics. Do they really trust it? Or are they going to go with what their gut tells them, or what as a clinician, what their experience tells them, their training tells them about our patient? Other people have done much better studies than I did, and looked in India at how people treat mock patients. So groups sent in mock patients with one of three conditions into various healthcare providers to see how they were handled, see how they were diagnosed. And I thought a peak of interest was they sent in a group of patients who were complaining of radiating chest pain that came and went, which is unstable angina, which could indicate you're about to have a heart attack. So for that, with these interactions, they found that diagnostics, which I've highlighted up at the top there, showed the percent of the interactions where that diagnostic was actually used with the patient. See, using a stethoscope to listen to the chest only got up to just under half the time they even checked that. Taking a pulse, less than that. Blood pressure, less than that. The tools that they did have available. An EKG, not all of them had an EKG, so I can understand that. But when people aren't using the diagnostics that they have through pressures of time, through a sense that they can diagnose, and that part of my skill as a provider is being able to just listen to what you say and make a direct diagnosis there. It's hard to overcome that. I don't have an answer, but I think it's something to be aware of. And yeah, another highlight from that same study, diagnosis being correct if they venture a diagnosis was only 9.5% of the time for those patients. And this isn't just in India, it's not just in certain parts of the world. I came back from my trip to Peru and had a really bad cough, and been to a number of TB clinics. And some of my coworkers were like, so, what kind of cough is that you've got? And my kids had been sick at the same time, had been diagnosed with pneumonia. So I ended up going to the doctor to see, and I explained all this. And he said, well, yeah, normally I would order a chest x-ray, but we don't have a chest x-ray. We don't have an x-ray technician right now, so let's just go and get you on antibiotics. It happens everywhere, right? There's a limited set of information. Every situation in some ways is a limited resource setting. Some a lot more limited than others.
Yeah, so the number that I'm seeing is interesting up there is that only 47% actually gave a diagnosis. So what, or 32% of all cases. So what did they do the rest of the time? They just say, oh, well, yeah, I guess you're sick. Sorry.
You know, I don't know. I'd have to go back and look at that paper again. And I'd be happy to send the slides out afterwards too, if you care to go and read up these papers afterwards. Yes?
The visit length actually says a lot about everything that was already discussed, because for me, for all the visits, seems to be under 5, except for asthma.
Right. Yeah, it's a shockingly short time.
So can you please clarify that number? What does it mean? Like 10% of the people who were diagnosed with this angina had it correctly, or I'm not clear, like, does it represent?
I shouldn't try to overstate my understanding of this paper. It's been a while since I worked through the whole thing. But I think the basic thing was, yeah, 90% of the time they gave a diagnosis that was not unstable angina. There is a partially correct category after that, which includes, I'm not sure what fell in there, partially correct. Another case, this paper is worth reading if you work in in vitro diagnostics. It was from Duke. I think it came out in 2013 about where they set up in 2 hospitals. It said for every patient who came in with fever, they let the hospital do their standard process with that patient, with whatever they wanted to do, drawing samples or not. And then separately from that, they said we'd like to collect blood samples and go off and do analysis to see what's there, see what might be causing this fever. So the Duke folks set up with CDC and with other top groups to do very high quality tests. They shipped off a lot of samples back to CDC to do so. They found that 870 patients that malaria was the clinical diagnosis for 60% of them. Clinical diagnosis, meaning that's what the clinicians there said they had and treated them accordingly. And it was actually found in 1.6% of the samples. So another thing to look out for. Right? We talk about that malaria is one of the most prevalent diseases around the world. And I think it really is. But do we really know? And how many times is it just assumed to be malaria? And how much antibiotic resistance is being promoted as a result of overprescription of antimalarials. Another important thing shown later on in this paper is 33% of the adults, and even more of the children, didn't match any of the things that they tested. What's making these people sick? What's giving them fevers? And finally, how different do you think things are in humans? Again, with my experience, with other experiences you might have had, how many times do you get a quick diagnosis that is the convenient one, but might not really be the right one? Number 5, life in the lab. So here's the Peruvian healthcare center that we visited. I mentioned Peru a lot because I was there most recently, so I guess it's fresh on my mind. And they had a pretty nice lab here. It was maybe a quarter the size of this room. It's not a major lab. They're set up to be able to try to handle their patient flow of 100 patients a day or so, which is a common size for a health lab. And we asked them about how do you do your TB smears? How do you do diagnosis for tuberculosis? As well, we collect all the sputum cups that we've gotten that come in from our referring sites. So those are all the tubs there. They're about 3 inches in diameter. And again, look at all those. One tub, one patient that might be transmitting the disease or getting worse. They collect all those and do their sputum processing for the day, which involves they take that burner that's in the middle there. And he reaches his hands around the burner as he's done, and he's going to smear the sputum. Like, take a stick, grabs the sputum out of the cup, and smear it on the slide. And the next one over. Because he wouldn't have the burner on, this was just a demonstration. Burner, what's the burner about? As near as we can tell, the idea is that the burner is supposed to capture the airflow that might be coming from TB, which is highly infectious, and take it up to the ceiling. So they have an exhaust fan in the room. They don't run it during the process, because they said that might actually draw more of the TB out. Then once they're done with it, then they run the exhaust fan. Where's the exhaust fan go? It just goes outside. It's right next to all the rooms. It's not really controlled anyway. A lot of places don't have any kind of airflow control at all. Sometimes they visit places where they do this on the top of the building, so that at least it's out more or less in the open, and then a little shed on the top of the building where they collect patient samples outside or something like this. But when you think of testing, you have to think of the safety of the operators, the laboratorians, and of other people who come into the clinic. The Ebola epidemic through last fall really brought that Ebola outbreak really brought that home too, as we saw. What was it? At one point, I think about 10% of the deaths in Africa were of healthcare providers being infected by the process of trying to treat patients. We've seen people in the U.S. who were having to watch and where laboratory processes failed and infection control measures weren't properly handled for Ebola here in the U.S. as well. So we've got to do things to protect the people who are working so hard to try to improve health outcomes. And telling them that you need to put this in a biosafety cap, a laminar flow bit, often just isn't enough, because if you don't have it and you can't get it, what are you going to do with it? Skipping a little bit to the Ebola story, there's some great papers that came out about this in the U.S. from Emory in Nebraska. One Emory showed how they set up their severe disease containment unit, which they've had in place for years. They sprung into action for that back last summer when the first 2 U.S. Patients arrived for treatment. They set up a point-of-care lab for all the tests for those patients. They made the choice not to have to send any of their tests into the main central labs because they couldn't be as comfortable or as confident of not spreading the disease through those ways. It's both something for peace of mind for the staff and for patients in the hospital, also for very real practical reasons. They weren't doing anything with the centrifuge because of the aerosol gases from spinning those down. They also avoided, except things they had to send to CDC, they avoided doing any transport outside the facility because they found that it's pretty hard to find somebody to take a suspected Ebola sample anywhere. Even the people who are supposed to be official shippers for those kinds of things, a lot of them just plain said no. So, reduced risk of transmission, faster results, they really liked having a point-of-care lab for that. So it led me to think, they didn't say this, but it led me to think, if it's good enough for US Ebola treatment at Emory, right next to the CDC, it's good enough for anybody. We get concerned sometimes that point-of-care tests aren't good enough. They aren't as high-performing as a central lab test. And that can be true. There are things that central lab tests can do that we cannot get to with point-of-care. But we sure can do a lot, and there are a lot of benefits to doing it. 6. Develops diagnostics in developing settings. So we see a lot of places when we go on these trips that look about like this that are very simple laboratories. In some cases, I think the sink over here showing a picture of left had no drain, had no running water, so hence the big bucket underneath the sink. Water be brought in each day for it. It works. It's fun across. You can see the pretty simple things in a long ways from what you see in many other places. A lot of sites are doing an awful lot of testing with a microscope and centrifuge and doing quite well with that. But you get the sense that that might be the last piece of equipment that they got quite a few years ago. But even if you go up to places that are relatively well-resourced, like this clinic in South Africa that was run by a study funded by Great Chalice Canada, you see that when they set that up and say, okay, let's do proper point-of-care testing with the best available equipment for this clinic with these 100 patients today. Well, in order to do it, for the variety of things they needed to test, they needed a gene expert, a Roche Reclitron, a HemoCube, and a PIMA for that whole battery of tests. And then they needed, pointers now, they needed this whole separate outbuilding to do that. They brought in a portable to be able to set up the test. Because it's not a lot of equipment, it's not a lot of space, but it's more than they could just fit into the health center. How are you supposed to do that without dislocating something else? And besides that, they also needed to provide a more regulated environment for these things. It needed to be dust protected, it needed to be air conditioned. So they ended up kind of creating this little bubble of a central lab, a little tiny central lab at the health clinic, not having something that was right there by the nurse's station or right where the typical patient flow was already happening. It's good, but it's not exactly point of care. It takes a lot more shuttling around, a lot more walking around to get samples there. And it's considerable infrastructure costs beyond just the instruments itself. You have to build this thing. Plus, we've got to deal with 4 different companies now for your service contracts and your consumables and everything else about the process. The training of the laboratorians, too, is different for every machine. So that brings us to PanDx. That brings us to the vision that we have for this, that we've developed at the leading guidance of the Gates Foundation. PanDx, we intend to bring high-performance, fast tests to the whole health centers, where patients, clinicians, and therapies are ready for them. Diagnostics are the missing piece there. We've got to make the results ready during the patient visit, have reliable results with minimal training, and have a broad menu of tests, including nucleic acids, immunoassays, and clinical chemistry, and really gold standard point-of-care performance. It's not about just making a test that we think is good enough for some other location or someplace. Nobody wants a test that somebody else thought was good enough for them. They want a test that's as good as you can get anywhere. A test that really works and that you would use for yourself or your family. And that's what's really missing too. A lot of tests do exist now, but they're not necessarily good tests, not strong tests. So with a strong test, we can get rid of the distrust in diagnostics to really make it worthwhile. So for PanDx, we say it's key for this to be easy to use, flexible, and robust. It needs to be fully automatic from sample in. Each cartridge in the system contains all of its own reagents, nothing to load, nothing to worry about. No biosafety cabinet required, and no computer experience. You can run a cell phone, you can use the system. For doing a TB test, get a sample that I've got from the cartridge. The process really is as simple as, sorry, I forgot to make these out of there. The process really is as simple as shown up here. So take your TB cartridge, the cup starts out as a separate thing, but I've already snapped it on here. Give it to the patient for the patient to expectorate directly into the cup. Put a cap on the cup that has a mixer built into it. Snap the whole thing onto the cartridge, and then it's shown, put it in the instrument, away it goes. You don't have to do anything else. There's no waiting, no operating wall. Yes?
What is the typical cost of that cartridge if you were to mass produce it?
We'll get to that in just a minute. Good question. Nothing matters. I'll try to get more quick video. It doesn't maybe running over time here. Sorry about that. I'll try not to go too late. It's very important that a system like this be flexible too. We've seen a lot of different locations. And while we're trying to make an almost universal system, one size fits all does not really apply because of the different number of patients, different types of tests needed in different places. But it is really important this is a random access system. So every single drawer in the system is independent, able to run any test that you might need and at any time. So if there's a free drawer in the system, you can run your test and treat that patient now. And it's critical that it be robust. We know the kind of places that these go into. We know that there's not someone coming around to do regular service on the machines. It's hard to get a replacement to something. So it's got to be well designed around proven technologies. We're not trying to go for anything all that cutting edge on this product really. We want to go with things that we know will work. It's got to be sealed against dust and water. That's a big one. It's something that often when you take a diagnostic intended for the US or Europe and just try to relaunch it over in Africa, but it's very difficult to deal with all the dust that's everywhere. A lot of clinics that we've been to don't have true windows, don't have glazings over the windows, just have openings. In Peru, I started to keep a log of pictures of people sweeping. Everywhere you go, there's someone working on that dust in the desert. So about the cartridges and about the cost. So this platform is able to run a broad variety of tests. We've been working on doing demonstrations of 3 tests, this tuberculosis test, as well as pictured up there with HIV-P24 immunoassay and ALT clinical chemistry test. So all 3 of these can run in our one platform that we have today. And I will pass these around. I would like to get these back, please. Start these. I'm not sure if you've seen this later. And then HIV viral load we consider as a design study to make sure that we could address it, but we're not building a demonstration of it right now. But it's such a critical test in global health that we had to address it. And it really requires a very large sample volume. This is the first thing, the assay types and sample types. There's a lot of exciting with very, very small samples. And it's exciting to see that. And yet, for some tests, you need a big sample. As Pete pointed out to us when we were talking about the sputum test we're developing here, you can't really ask a patient to only produce 1 milliliter or a few, a certain number of microliters of sputum. When it comes up, it comes up. You've got to do something with it. So we've got a big cup to be able to accommodate that. And bigger samples do mean better precision too. And that's part of what limited point of care tests for their quality. So if you go into a central lab and you get that venipuncture draw, you pull 5 milliliters of blood, they can do a more accurate test that way often. So for some tests like HIV viral load, we still believe that we need to do that. So we designed the whole system around being able to process those big samples. Yeah.
Are these meant to be reused?
No, they're all one-time use cartridges. So with having preloaded reagents, everything it comes, the device is just,
Nice. Thank you.
Yes.
Are you going to publish something that's sort of, I guess, equivalent, like a hardware equivalent API, so other people can develop tests on it or not?
It's a good question. There's a lot of talk about that early on. Be careful not to go on too long without it right now in the talk, but there are benefits to that clearly. The potential disadvantages of control in terms of ensuring that a good test is there. So with a so-called laboratory developed test, how do you make sure that it's actually a good test that works well? So that there are a lot of pressures actually against them in this part. To instead say, let's make sure we deliver a test that is that we can stand behind and we have a good result for.
And how have you been thinking about disposal of the medical waste?
Disposal is
Because these are fairly significant, infectious,
That's right.
and reusable potentially.
Potentially reusable. People love to reuse things for anything they can. When we talked about concepts one time, someone said, "Oh, people are going to want to turn these into doormats or something. They string them together and turn them into something." For an earlier style of cartridge concept, which it's very hard to predict how people will want to use something. So they'll be designed very much so that they cannot be reused. So they will not provide any kind of result afterwards. The machine even can recognize that the cartridge has already been used. It's just they wouldn't be strictly used in that way. The disposal problem, I don't have a great answer to, frankly. We would love to have something that's a biodegradable material, these cartridges. But on the other hand, we want a cartridge that can be stable at 40 degrees C for 2 years and perform in these ways that are quite challenging overall. So we don't have the materials yet that can serve that. So ultimately it comes down to incineration still for these things. It's a safe disposal method. And sampling and activation is part of the strategy. So that as much as you can within a test, try to make it non-infectious by the time you process it. I wish I had a better answer, but that's the partial answer.
Yeah, it's a big problem that very few people have solved. All the cartridges now that are going are just, a lot of them are just getting chucked out the back window or the back door.
Yeah, and we've seen pictures of the sites that have an incinerator with a fireplace chimney kind of thing, but they haven't been used in months. There's just a big pile of cartridges. There's just time, I won't go too much more through the things on this table, but yes?
Quick question. So, I guess how much is your machine cost that this little each sample use for like the big machine? And I guess who buys those machines? I mean, I'm assuming that since developed countries they don't have as much, I guess, money compared to the United States. So I guess who buys those? And I guess how can they make sure they can maintain the machine? And I mean, I don't know. And also the kind of, I guess, how much do you guys sell it so that if you get, because you guys are a company, you have to run it with profit, right? So I guess I'm not sure how you guys kind of find the right niche that you guys get profit at the same time you guys are selling at a low cost to the developing countries so they can use it. I don't know if I didn't know it's I didn't mean to be a very long question, but
Yeah, there are a few good questions in there, important to the whole scheme of making something like this successful. So first, Stratos is a design group, we're not a manufacturer, actually. So we work directly with the foundation to come up with the designs for it. We routinely work to then turn those into products and get them into contract manufacturing situations for our clients. In this case, the more likely route would be the foundation would work with a major diagnostics company. We're in talks with a number of those about pursuing such a path. So, but as to the economics of it, the approach that we're using is to focus on making it as cheap and high-performing and disposable as we can. And the business is a lot like the inkjet printer business, really, in that you may think about the initial purchase price of the machine, but where the revenue comes from for the manufacturer is the disposables. So for a given system, you can easily be running 1,000 tests per bay, and we're showing a model with 5 bays per instrument. The costs there are huge. It generally works out to that 90% of the costs come into the consumable rather than the equipment. that's our focus. We're trying to do everything we can to keep that down, keep it low. Even where we are today, the goals are to be less than $5 for advanced tests, for advanced nucleic acid tests like a TB diagnostic that we're doing, and to be considerably less for other tests that are already cheaper today.
Sorry, how do you handle this? I think many countries would kind of have some duty for import of these kinds of things from probably if this is getting manufactured, say in China or maybe here, if you have to ship them to countries which are developing, those countries might have some regulations against import of foreign products, right?
Right.
So does this also account for those expenses or I don't know?
Well, so when we talk about cost here, we list cost of goods sold. So it's not including tariff, not including profit, not including distribution. So it's strictly making this cartridge and having it ready for delivery to somewhere. So it's a nice level playing field when you're talking about engineering developments to talk in terms of costs. But it's not inclusive of all those other things. And we've seen that as well. We're very familiar with that. It brings up the questions of being able to build either instruments or devices within countries to avoid some of that, to get greater buy-in and support. But that's a complicated business too. So how do we do all this? How are we running so many different assays on one instrument? The basic idea is to provide a lot of different services, provide a good suite of all the capabilities that you might need, and then let each cartridge pick from it and use the ones that it needs. So for our tuberculosis cartridge, we're using a lot of those interfaces. For our HPV load cartridge that we would do, we aren't building that. We use a slightly different set. We need to do reverse transcription as well for that assay. For P24 antigen, we're actually in our current implementation of it, showing a pretty complex full ELISA. It includes a 3-step labeling of it, so we use quite a few of our services to do that as well. And then for a simple test, like an ALT clinical chemistry test that we do with a dry reagent pad, it's a much simpler process. You can actually make a small cartridge. This was an early rendering of the size of cartridge we might use. With what we have today, we could actually take that full-size cartridge and cut away most of that plastic and get down to a reduced-size cartridge. I have quite a few more slides that I'm going to skip past, but we're already in time and I want to get to more questions. I'll just give a brief tour to come up with this. We went through an in-depth process of looking at all the processes that we needed and the different ways we could provide those services to an assay. Anytime you develop a diagnostic system, the tail wags the dog if the tail is the assay. So you've got to design around what the assay actually needs, make a cartridge that works well for that, and make an instrument that works well for that. So typically for us, the assay is going to be first, it's going to be most mature, and the cartridge goes second behind that. And then to a degree, the instrument has to live with whatever the other two can do. It has to be able to support them well. So we looked at those interfaces. I can't go into detail tonight, we looked at the interfaces electronically as well, how important it is to be able to provide data to clinicians there at the site, as well as epidemiologists and others doing research. And we looked at all the tests that were on the wish list, and the wish list changes a lot. But it comes back to certain key categories. We've tried to describe all those categories, and we did an analysis to show that, yes, we could run this type of test for every one of these tests on our system. We believe that gives a good standpoint for saying, yes, we can run new tests that come up over time, which is a key consideration for launching one. We mapped out the processes you use for these tests and the way we'd actually do them. I'm trying to go really fast now to catch up. And we looked at lots of different hardware concepts for dealing with this, from very small instruments to the bigger things we've shown now. From things where you dedicated, limited performance in smaller sizes, things where you use different adapters to be able to run small, simple tests on bigger platforms. Different configurations, all around this focus of let's make a powerful bay. Let's talk about how we can package it to be most useful to people. We ended up finding with a lot of this, with different tiers of hardware and that sort of thing, trying to make something that was a little bit cheaper, a little bit smaller for certain purposes. A lot of people got confused and got back to the point where they said, what is this for? How do I use this all over? So we ultimately came back to, we've got one slot that can do everything on every instrument. This is pretty simple. Again, because the costs really come with the disposable, it wasn't worth it from our analysis to break it up into different products. There's of course still an interest in having a truly portable product, which this is not. This is a benchtop product that stays stationary within the facility. And you can see where you can take this product and work towards with other technologies, making something that's handheld and serves all those needs. Our development steps, we're doing an 18-month project now to show technical feasibility of the system. So we've been building breadboard systems and building these cartridges that you see right here. Microfluidic chip shop in Jena, Germany, headed by Holger Becker and Claudia Gardner, who I understood spoke here just a few weeks ago, are our collaborators in this. We chose them for doing the assay cartridge work on this project. And we go out to Jena regularly to work with them. It's been a very good relationship. Here's the breadboard instrument that we built at Stratos to be able to do this. It looks nothing like a production instrument, but it works very much like a production instrument. If you were a cartridge, you could go inside that instrument. You see the same things, you experience the same things that the cartridge would experience in a production instrument. So this let us build the system quickly and modify it quickly so that we can prove that it works, and so that we know really what the requirements are for each part of the system. How well do we need to control the position of the things that push down the blisters? How well do we need to regulate the temperature to really get the results that we want to get? So that is a whole next project. We then take that understanding of those requirements and translate those into mass-producible designs that hit the cost targets and reliability targets. And I wanted to bring at least a few slides of data just so you knew we were doing something more than just talking about this, making pretty pictures and that sort of thing. It's still preliminary data. What we're working towards is by early this fall, September, having data from hundreds of cartridges for each of our assays. Skip that actually. Hundreds of cartridges from each of our assays run with spiked samples. Not yet working with real clinical samples, but going through the process of getting it working well with known controlled samples for each of these to show that yes, you really can do these three very different assays on one instrument. So at that point, we'll consider that the major technical feasibility has been demonstrated and we're ready to move into commercial product development, which is actually where Stratos spends a lot of our time on other projects, taking things all the way through to manufacturing. One last story from the field. Not my story, but if you go back 50 years to the smallpox eradication campaigns, two tools were really key in delivering those vaccines. The one on the left, the jet injector. They made a special version that was foot operated. It was basically something that had been developed by the Army and used without a lot of consideration to cost. It had been around for 20 years prior to that. Worked pretty well. Took a good amount of maintenance. It used a lot of vaccine with every shot. The device on the right, bifurcated needle, is shown gigantic, but actually the whole thing is just a few inches long. Those tips are just far enough where you can dip it into some vaccine and it holds a little bit of the vaccine, the right amount of vaccine, so that you can then go and poke somebody's arm with it to get a take. The design on the right there is a beautifully elegant and simple design. Very low cost, very easy to sterilize, and used, I think, a 40th of the vaccine per patient, as the jet injector did. But they didn't have it when they started campaign. They didn't wait for it. They kept doing research. People came up with it. But they got more than 5 years in through using the jet injector and had vaccinated thousands of people and been able to remove the virus from a great number of places. The point being, it's great that we work on technologies for someday, but let's also use technologies we have today and use them well. I think for diagnostics, there are a lot of technologies that are already in the field, many of them off patent, that can be used well, but just haven't been implemented for this certain application. So that's been our focus, is to try to take things as quickly as possible, make it into a good product that can serve a lot of purposes, while people here at Berkeley and research universities around the world and startup companies continue to come up with the next great wave of things to come after. So we're excited about your work, and we're hoping you're excited about our work as well. This story comes up in a book by Bill Fahey that I highly recommend to anybody working in global health. Bill Fahey was one of the key people in the smallpox eradication campaign, and has been an advisor for the Gates Foundation as well. House on Fire is the book that's written for a popular audience. It has lots of great stories, more of a memoir than anything. Many hands-on stories. And he's got a great attitude, very positive guy. He talks about the need for optimism on his teams. So we try to keep that in mind too. And I won't read the whole quote, but I do laugh a little bit. Being a consultant, and like Pete, it says, there's a place for cynicism, a place for pessimism. Contract for it when you need it, but don't get those people on your payroll. They will ruin your day. As a consultant, I wonder where I am in that. So I'd like to recognize everybody who's contributed to this project, especially the Bill and Melinda Gates Foundation, not only for funding our project, but really having the vision for it, for taking an ambitious but practical goal and driving towards this and being very engaged with us week in and week out on how we develop this. It's been valuable to us, great education, and we're very thankful for the opportunity to contribute this way. And I'll leave it at that. Thank you very much. Yes?
Yeah, so how's the reception been from the public health community on this work? Because talking to a lot of public health people, it seems like some of the enthusiasm for technology-based solutions has kind of gone down in recent years, and a lot of that has to do with what you mentioned about some of these donations of high-tech solutions that just can't be implemented in these different areas. So people seem to be going more toward, sort of structural society-based interventions rather than technology-based interventions. So if you had any pushback for people that feel about technology-based solutions?
Yeah, that's a good point. There's always a trade being made in where you spend the dollars. Do you spend it on the better diagnostic, or do you spend it on the healthcare worker who can go out and make rounds and make sure that the person they already know does have a disease is actually being tended to and being taken care of. I'd say that it's not an either or thing. We haven't talked with any public health group that sees it strongly as one thing or another. They are doing diagnostics. They have to do diagnostics. You have to know if the person has tuberculosis or HIV or malaria to be able to treat them well. So the tough part is how much do you spend each place. Anything else? Gotta be more questions. Did I really go that long? It's just time to go home.
How much is actually the analyzer cost? The analyzer?
Yeah, I didn't directly address that before either. So we're doing it in terms of the columns, again, cost of goods sold analysis. And what we've come to is roughly $2,000 per drawer of the instrument when you break it down that way. So it's not cheap. We would like it to be cheaper. There's some ways we could make it cheaper. And as we go forward, we'll be investigating those both from a manufacturing and design standpoint. So we're trying to be realistic in our estimates. We're trying to be so far. And again, it works out with those numbers in a lot of ways, given how much testing you can do on a platform like this. Really that's what the cost is.
But what is the major cost also? Is it the reagent, the testing, the robotics of it, or other parts?
Within the cartridge?
Yes.
So for the cartridges, I can't make public the numbers that we're using for that. You can see some other analyses. I encourage you to look for, there's an analysis that Fund did a couple of years ago, the HIV viral load tests, where they independently assessed what they think the costs were for several different systems during production. And they break that down according to the materials and reagents and so forth. That's probably the best place I would go. And Cepheid's actually made some public slides too about portions of their costs. At least they go to different places for the TV market.
Why did you choose the particular assays that you did for your first round?
Yes, we wanted to show breadth more than anything. We knew we had to show a nucleic acid test for the application. We had to show an immunoassay, and then it'd be nice to show a clinical chemistry assay too, to show something different, show a little breadth there. For the particular assays, we needed to find ones that we could do as a demonstration on this project. We're not doing full-on commercial development of these assays yet. We didn't have time or budget to do that in a way that really needs to be done. But at this point, we're still at technical feasibility of the system. So we said, and we had very close collaboration with the Gates Foundation on this. So let's just show something that's challenging and that people understand, and we can work with that. So tuberculosis obviously fits in quite well. There's a real clinical need for that, and a lot of people appreciate that that's not an easy test to run at the level. HIV-B24 we picked because it is a complex immunoassay that's done today in central labs. You might not do it when you're actually at the point of care. You might skip the viral load test instead, right? Nucleic acid detection test instead. You're still going to need other immunoassays. So we said, here's one that people understand. It is a global health concern, rather than us giving some target that has nothing to do with people in this area. Does that make sense?
Yeah. Well, you could have approached it in a number of ways now. That's an interesting choice to do it that way. I mean, it's a good choice. And a secondary question on that. Do you have a sense of you had a menu that you went through, you bypassed quickly for time. How much time do you have any sense of, per assay, an average time for developing the clinical capacity? Obviously, you're demonstrating that the platform can manage to be able to run those kinds of tests, then the question is how long would it take to develop those?
Pete, would you care to address that?
If it was a nucleic acid test, I mean, a good, going for like European or US approval, typically 2 to 3 years for a nucleic acid test, shorter for amino acid test. And I'm not really sure about the chemistry test, probably shorter, but it's not much. It's significant.
Right, it's significant.
Yeah, the whole next phase of this project, if the Gates Foundation approves it, would be to develop real assets for direct clinical applications and production instrument for that. It can also be a little bit less time than that, but similar time. So we are working 2 to 3 years on.
If working with an industry partner who already has all of these assays on another instrument, it's still going to take time, but it will be faster.
And we've worked hard to try to accommodate many initial processes. That way, so that really convert an assay rather than building from scratch.
Right. That was really where I was going.
Sort of a general question. Where do you envision point of care 5-10 years from now? Do you think there's like unvisited turf that's like really important?
What was the last part?
Is there turf that's unvisited?
Is there a what?
Is there unvisited turf?
Turf. You can kind of joke in microfluidics and molecular diagnostics that it's the next big thing. It has been for a decade. So I don't know. I think anybody who has very clear visions about that with a grain of salt, I'm not really sure. But we see, I think there are places where the technology has become a lot more mature in the last few years, and where we were able to put together this functional system in less than 18 months to where we are now by using a lot of things that have been proven out over the years and skills have been developed. So it's very feasible now that in the US, the payment system and FDA regulations about it make it difficult. FDA requirements that in order to get CLIA waiver, you have to show that you were as good as the predicate device, as good as a single lab device, whether that's actually needed or not. And we have very strong safeguards against possibly displaying an erroneous result, making it quite challenging for that part. So it's one of those things where you could say maybe it's going to be another leapfrog situation where places outside of the US fully embrace point-of-care testing sooner than we do because of all these legacy systems and structures that we have set up.
So your current system that you guys have, I guess what is it? Who is your exact target? So are you trying to target the like rural areas in the developing countries where there's like, you know, not like really private hospital, but there's where they're like very rural hospital and there's very limited amount of machines there? Are you trying to implement your device to there, or are you trying to more first try it out in advanced or like I guess like private hospitals and then trying to go down to rural hospitals later? I guess I'm not clear about what's your target output. I guess what's your target hospital is first, and then
Yeah, I think I didn't explain that quite enough tonight. Sorry about that. But I did mention about level 0 and level 1. We often see these pyramids where level 0 is the level, very base level of the healthcare system, often involving workers who have some training but who are not doctors or nurses, to be able to check in with patients and that sort of thing, check with people who might be sick and refer them to care, or they staff a small post, but there's no one there to actually treat you directly. If you have something, they'll refer you to another place. Our target is just that very first level above that, to the primary care level in developing countries, similar to the primary care level here and the urgent care clinic level that we have here. Places that patients naturally go today, which look different in different countries. Some places that's predominantly public, some places it's predominantly private. Either way, there's that level where patients are going and being seen for the first time without having to be referred up higher than that, trying to help avoid those patients having to be referred up or lost out of the system. So frequently it's a clinic that has 50 to 200 patients a day. We've seen ones that are actually quite a bit bigger than that. We've seen some in South Africa that are close to 1,000 patients a day, but still act as primary centers.
That's all. Thank you for coming.